COVID, reactivated herpes viruses, HERVs, human endogenous retroviruses, and Behçet's disease, Part 1. This is the first part of a multi-part discussion on this topic. The goal of this particular YouTube series is to examine how COVID infections can reactivate a set of dormant pathogenic agents in tissues. And in this YouTube series, we will discuss how this can take place in an autoimmune condition known as Behçet's disease. We will also examine the utility of vitamin D receptor agonists as a means of suppressing one of the key upstream drivers of tissue pathogenesis, and that is a herpes virus, HSV1, herpes simplex virus 1. To set up the background for this discussion, I'd like to first emphasize a key paper which was published early in the COVID pandemic. Human herpes virus 6, human herpes virus 7, and Epstein-Barr virus reactivation in pityriasis rosea during COVID-19. Pityriasis rosea is a type of rash. In this case, it was a young man who developed a rash-like set of lesions in the back of his throat. And when these lesions were examined, they found reactivated herpes virus 6, 7, and Epstein-Barr virus reactivation in these lesions. Now, if you look on PubMed for a different human herpes virus, it's human herpes virus 1, which is known as herpes simplex virus 1, HSV1. If you look for HSV1 in Alzheimer's, you'll find 325 results. This is because HSV1 is now recognized as a potential or key driver of Alzheimer's. Now, in the bottom part of this slide, I'd like to discuss certain types of lesions in the oral cavity, gingival, which refers to the gums, gingival recessions. And this is where the gums are receding from the teeth, and these recessions are caused by herpes simplex virus in a patient with COVID-19 infection. So this emphasizes that COVID, by reactivating a herpes simplex virus, initiates lesions that are characteristic of the secondary infection. And that's using an epistemology which views the COVID-2 infection as the primary agent and the HSV1 as a secondary event. In this YouTube, we will now discuss the ramifications of HSV1 reactivation, particularly in the reactivation of human endogenous retroviruses, HERVs. To discuss these connections, as well as potential therapeutic avenues, I would first like to draw on this recent review, Transactivation of Human Endogenous Retroviruses, HERVs, by Viruses. What does the term transactivation mean in this context? To start this discussion, it's useful to draw from AI Overview. AI Overview provides a concise but overly limited definition of viral transactivation. Viral transactivation is a process where viral proteins, such as HIV-1 TAT or adenovirus EA1, act as transcriptional activators to significantly increase the expression of viral or host genes, usually by recruiting host machinery to specific viral promoters. These proteins are essential for viral replication and often operate in the nucleus. In contrast, here is a definition of viral transactivation from Wikipedia. First, a discussion of natural transactivation. Transactivation can be triggered either by endogenous cellular or viral proteins, also called transactivators. These protein factors act in trans, i.e. intermolecularly. HIV and HTLV are just two of the many viruses that encode transactivators to enhance viral gene expression. These transactivators can also be linked to cancer if they start interacting with and increasing expression of cellular proto-oncogenes. HTLV, for instance, has been associated with causing leukemia primarily through this process. Its transactivator, TAX, can interact with P40, inducing overexpression of interleukin-2, a cytokine, interleukin receptors, GM, CSF, and the transcription factor CFOS. HTLV infects T cells, and via the increased expression of these stimulatory cytokines and transcription factors, leads to uncontrolled proliferation of T cells and hence lymphoma. Artificial transactivation. This is the type of transactivation that cell and developmental biologists are usually thinking about. Artificial transactivation of a gene is activated by inserting it into the genome at the appropriate area as transactivator gene adjoined to specific promoter regions of DNA. The transactivator gene expresses a transcription factor that binds to specific promoter region of DNA. By binding to the promoter region of a gene, the transcription factor causes that gene to be expressed. Let us return to the review published in 2024, Transactivation of Human Endogenous Retroviruses by Viruses. Now in this wheel diagram, in the center of it, you can see certain herpes viruses such as HSV1 and Epstein-Barr virus. On the right, you see SARS-CoV-2. Reading the figure caption, summary of HERV activation by viruses. Human endogenous retroviruses that are activated by the indicated viruses and virus families are listed along with the types of samples where HERVs are detected. The post-viral diseases that are associated with the viruses are also highlighted. These disease states are highlighted by the outer rim of this wheel diagram. If you'll look to the right, you see a code for this information, associated post-viral diseases, neurological symptoms, multiple sclerosis, cancers, type 1 diabetes, hepatic disease. On the left, I have highlighted a specific herpes virus, HSV1, which we will discuss further in this YouTube series. HSV1 is capable of transactivating HERV-K protein as well as HERV-W protein. These retrovirus proteins are pathologic, and they are known drivers, contributors to the following disease states: multiple sclerosis, MS, amyotrophic lateral sclerosis, ALS, Alzheimer's disease, AD, Parkinson's disease, PD, rheumatoid arthritis, RA, and long COVID. Now, adding CoV-2 to this complex jigsaw, and that is a term that British people use to refer to as a jigsaw puzzle. Looking at the various pathologic agents, whether they are proteins or viruses or pathways that become unbalanced because of these agents, these are all part of the jigsaw that produces these disease states. And this creates a challenge for the epistemology of pathogenesis, especially histopathogenesis. For instance, the issue of a HERV protein. Is it a host factor? Now that might seem to be a strange way to refer to a fossil virus, which has been incorporated into the human genome. But that is the crux of the matter. These HERV viruses are present in all human beings, and they're pathologic when they become reactivated. So why do some individuals have many decades of life without expressing the pathogenesis from these viruses, these fossil viruses, HERV viruses? What creates susceptibility for tissue pathogenesis? These are some of the key issues that need to be addressed, because CoV-2, by reactivating HSV1, reactivates HERV-K protein expression as well as HERV-W protein. And on the right, you can see that CoV-2 by itself, independent of HSV1, can do this. So this is a double hit system where CoV-2 can reactivate these HERV viruses, their proteins, which are pathologic. In addition, it can augment the pathogenesis by reactivating HSV1, which in tandem with CoV-2 expresses these HERV proteins. So what are the transactivators of HERVs in long COVID? Now I would like to connect to a separate YouTube series on this channel where we discussed the hypermethylation of the GPX4 promoter. Recall that GPX4 is the acronym, the abbreviation for glutathione peroxidase 4. This enzyme utilizes glutathione to reduce peroxidated lipids as well as reactive oxygen species in cells that are undergoing oxidative stress. For instance, a viral infection or a therapeutic treatment such as hyperbaric oxygen. So methylation of the GPX4 promoter is potentially very pathologic, and it is occurring in long COVID and in ischemic reperfusion injuries. In addition, the vitamin D receptor, its promoter, can also become hypermethylated in infectious diseases such as COVID, and it can also become methylated in autoimmune diseases and cancer. So losing antioxidant capacity sets up tissues for increased lipid peroxidation, which are drivers of the programmed cell death pathway known as ferroptosis. So ferroptosis is a key element of this jigsaw puzzle. It is a pathologic process by which many tissues and blood cells can be damaged or eliminated as a consequence of viral infections or perhaps transactivations of HERV viruses in these pathological states. And this is pertinent to specific disorders such as Alzheimer's disease. So HSV1 in Alzheimer's disease, several recent references. HSV1 is a potential driver of Alzheimer's disease, unveiling the involvement of herpes simplex virus 1 in Alzheimer's disease, possible mechanisms and therapeutic implications. Role of HSV1 in Alzheimer's disease pathogenesis, a challenge for novel preventive therapeutic strategies. And on this YouTube channel, we have discussed the role of HSV1 and COVID, and a URL link to that YouTube is highlighted in green at the bottom of the slide. So we will draw from this literature and examine the cellular, molecular, and tissue aspects of pathogenesis in these neurodegenerative disorders and how the upstream drivers, particularly the reactivation of herpes simplex virus 1 and the downstream transactivation of HERV-K protein or HERV-W protein, contribute to the tissue and cellular pathogenesis which typifies Alzheimer's disease. Now, before proceeding with the rest of this particular YouTube, I would like to introduce this paper from 2010. I believe it is very important. It's one of the few papers on PubMed that illustrates that herpes simplex virus 1 can infect other cells in a nerve that are not neurons, glial cells. And one type of glial cell, which is highly neglected in terms of being a reservoir for HSV1, are cells of the perineurium. So what is the perineurium? The perineurium is a connective tissue sheet surrounding a nerve fascicle, which I will define in a moment. So this tissue sheet is composed of cells known as perineurial cells, and HSV infects perineurial glial cells. So if you look in the upper right of this slide, you'll see an exploded diagram of the structure of a peripheral nerve, such as a cranial nerve. An example would be the vagus nerve or the trigeminal nerve. These are cranial nerves, but they are part of the peripheral nervous system. And it is well known that HSV1 infects the trigeminal nerve and infects the cells of the neurons of the trigeminal nerve, which are located in the trigeminal nucleus. That's the standard model. Unfortunately, when you have standard models, that becomes dogma. And dogma is when people say, I've never heard of HSV infecting glial cells. It must be incorrect because I haven't heard about it. And this is one of the problems of translating preclinical models, example, rodent models of HSV1 infection, into neurology and psychiatry and now immunology, because this paper from 2010 demonstrates that perineurial cells can be infected by HSV1, and the perineurium is a site from which HSV1 can reactivate. Let me read from both the beginning and end of this long abstract. Herpes simplex encephalitis, HSE, is a rare disease with high mortality and significant morbidity among survivors. We have previously shown that susceptibility to herpes simplex encephalitis was host strain dependent, as severe lethal herpes simplex encephalitis developed after injection of human herpes simplex type 1 virus, HSV1, into the whisker area of DA rats. That's a type of rodent strain, where PVG rats, a different strain of rats, remain completely asymptomatic. In the present study, we investigated the early immunokinetics in these strains of rodents to address the underlying molecular mechanisms for the observed difference. Now moving to the end. Our data demonstrate the novel observation that genetically encoded properties of the host nerve and perineurial cells, recruitment of phagocyting cells, these are white blood cells like macrophages, together with the low expression of TLR2 and TLR9, these are toll-like receptors, in the periphery define the susceptibility to HSV1 entry into the nervous system. Now, in the upper right, you see an exploded view of a peripheral nerve. And peripheral nerves include cranial nerves. There are 12 pairs of cranial nerves in vertebrates. One pair, there is a right and left vagus nerve. If you look at a cross-section of a peripheral nerve, you'll see that it contains bundles, and these are called fascicles. The term fascicle is derived from the Latin term fasces, which is a term for a bundle. Here we're talking about a bundle of axons, which are myelinated by Schwann cells, and that is a term used in neurobiology to refer to a wrapping around of an axon by a glial cell, and this is called a glial sheath or myelination. The purpose of it is to increase electrical conduction and to prevent electrical crosstalk between adjacent axons. Now, the outer portion of this fascicle, this bundle of axons, is a connective tissue sheet, and that is the perineurium. Between axons, inside this fascicle, there is an endoneurium. So we have endoneurium adjacent to the axons. All of that is encompassed by this sheath, illustrated with a black line. And these fascicles are then wrapped in an epineurium, and that is the outer part of the nerve, thus the term epi, to the outside. This paper discusses infection of the perineurial cells. They're part of the perineurium, and the roles of toll-like receptor 2 and 9 on the susceptibility of these cells to HSV1 entry and infection. Reactivation from this site in one of these rodent strains, rat strains, was found to make the rat strain susceptible for the infection to spread into the central nervous system, leading to herpes simplex encephalitis. Reactivation of herpes simplex virus 1, either from the trigeminal ganglion or from perineurial cells in the trigeminal nerve, can lead to the production of infectious virions, which can move through axonal transport either into the central nervous system or out to peripheral organs. And it is well known that HSV1, once it reactivates, can move to the oropharyngeal cavity and produce cold sores, also on the lips. And in other cases, HSV1 can move to the eye and infect the cornea. This is called HSV1 infectious keratitis. So at the bottom of the slide are two papers that pertain to this. One is a novel use of vitamin D receptor agonists to treat an HSV1 infectious keratitis. It was a severe case. The cornea perforated, which allowed the iris to protrude from the cornea. Very dangerous situation for the eye. The patient was hospitalized, given standard of care acyclovir to suppress the HSV1 infection. A graft of an amniotic, fetal amniotic membrane was grafted to the eye. That was also standard of care. When the patient was discharged, the HSV1 reactivated again, but throughout the body. So IV acyclovir was also given, an outpatient. But to bolster the innate and adaptive immune systems to help suppress HSV1, the patient was treated not with vitamin D3, cholecalciferol, but hydroxylated versions of it. 25 vitamin D3, which is calcifediol, and 125 vitamin D2, paracalcitol, a synthetic analog of calcitriol. And utilizing calcifediol, paracalcitol as adjunctive therapies, the infection was suppressed. The patient ended up with a corneal scar, a cataract. However, after a year of being maintained on calcifidiol and pericalcitol therapy, the patient was able to undergo a corneal graft, and that, of course, removes the cataract because a new cornea has been grafted to the eye. And remarkably, the patient has now 80% normal vision in that eye. So I bring this up because the paper on the right, labeled 3, shows oral ulcers in an autoimmune disorder known as Behçet's disease. And this is common in a group of ethnic groups in the Middle East which have a specific HLA haplotype. So in Behçet's disease, oral ulcers are common. They become activated, and then with treatment they often go into remission. But in this patient, the patient had a flare of these oral ulcers as a sequelae to COVID. Fortunately, they were able to treat this woman, and I will go into the details of this case study in a subsequent YouTube. She was treated with vitamin D3. Her calcifidiol level, or 25 vitamin D level in her blood, which what people call vitamin D level, was 9 nanograms per mil, very low. She was immunodeficient in this critical immune hormone. By bolstering her 25D levels, boosting her, quote, vitamin D levels in her serum, the oral ulcers went into remission. Now, we will talk about these oral ulcers in Behçet's disease because they are frequently infected with HSV1, making it plausible or even likely that HSV1 is one of the key drivers of this ulceration. Now, how does this pertain to COVID? In 2020, a group in Croatia published this key paper, a theoretical paper. SARS-CoV-2 dissemination through peripheral nerves explains multiple organ injury. And we now know that COVID-2 can be transported within endosomes within the nervous system. And so when mucosal tissues like the lungs, or the tissues that line the lungs and the gut, become infected with COVID, the infection can be amplified there, and virions can move from the mucosal tissues into neurons that innervate these mucosal layers of these visceral tissues. From there, the infection can spread through these peripheral nerves up into the brainstem. And so this leads to persistent viral infections, as well as reactivation of herpes viruses that lie in these same nerves and brainstem. And I'd like to emphasize that one of the potential reservoirs for HSV1, again, is the perineurium. And so in the case of COVID and long COVID, we really need to consider perineurial cells as a place where HSV1 can reactivate, and where these human endogenous retroviruses, the herpes viruses, can also reactivate the perineurium. And so here, bringing these elements together, SARS-CoV-2 entry sites are present in all structural elements of the human glossopharyngeal and vagal nerves, clinical implications. And this diagram helps illustrate that you have an organ, in this case the tongue, or the pharynx, or the epiglottis, and these portions of the oropharyngeal cavity and its organs are innervated by different cranial nerves. The 7th, the 9th, the 10th, and each of these nerves has a bundle of cell bodies, and these are known as ganglia. The geniculate ganglion, the petrosal ganglion, the nodose ganglion, and that's part of the vagus nerve. these neurons project into the brainstem, and they influence the dynamics of second-order neurons present in the brainstem. That's how signals are transmitted, relayed to the brainstem, and from there to higher centers. This group of second-order neurons are involved in autonomic functions such as breathing rhythms. For instance, the Butzinger complex and the pre-Butzinger complex. So viral infections, if they're transmitted into the brainstem, this can have severe pathologic damage to the brainstem, or just neuroinflammation of the brainstem can alter the function, the autonomic functions that are controlled by these regions. And this is one of the ways that dysautonomias can be established. Now, this paper published in 2022 was solely focused on SARS-CoV-2, not the reactivation of herpes viruses or the reactivation of HER viruses, which are both possible as a consequence of a COVID-2 infection moving through cranial nerves into the brainstem. So we're going to take up that particular issue, this pyramid of pathology emanating from a primary COVID-2 infection linked to a secondary HSV1 reactivation to a tertiary reactivation of HER viruses and the expression of these pathologic proteins involved in neurodegeneration. Now, in part 2 of this discussion, we're going to take up this case series, well, it's a set of case studies. Clinical activity of Behçet's disease associated with SARS-CoV-2 and herpes simplex virus. Insights from 3 case reports. This is published in 2025. Finally, in closing, I would like to encourage you to visit other YouTubes on this channel. Here is a series that discusses ferroptosis driven by the silencing of the gene which encodes GPX4, glutathione peroxidase 4. And this silencing is mediated by hypermethylation of the GPX4 promoter. And this paper in particular was discussed as a central mechanism by which injuries that occur during COVID and long COVID, which result in ischemic reperfusion injury, could lead to the hypermethylation of the GPX4 promoter, silencing this gene, which is critical for antioxidant, anti-ferroptosis capacity of cells. So if you would like a PDF slide summary of this talk, as well as other talks on this channel, these PDF slide summaries contain active links to YouTubes as well as references on PubMed. Please go to the folder highlighted at the bottom of the slide, COVID therapies. Type in this URL. It'll take you to a Google Docs folder where you can find a set of folders. If you'll navigate to the folder download first, you can find the PDF slide summary to this talk more quickly. So I will pause here and then proceed to part 2 of this discussion.